Hello, welcome to the second experiment called the determination of enzyme specific activity. The objectives of these experiments are determine the Gst enzyme activity and determine the protein concentration by Biuret method, and the last one is determine the specific activity of Gst enzyme. The fundamental of these experiments are the activity of the Gst enzyme in a protein mixtures depends on the amounts of protein used in the reaction. Therefore, it is necessary to know that the protein content in the added volume. So as we can see here on the picture, there is a monomer and polymer here. So the monomer is a small molecule. Let's say this monomer contains two white circle and one black circle, while the polymer is a long chain molecule made up of repeated patterns of monomers. For example, here there are four repeated patterns of monomer containing white, and a black circle. So a monomer is a single molecule that can be joined together with other same molecules to form a polymer. The building blocks of a proteins are amino acid, which contain elements such as H, N, O, C, and more. They are the monomers of the proteins. When hundreds or thousands of amino acids join together, they create a proteins, which are then used for many tasks in organisms, such as doing work in cells, help with DNA replication, and so on. So we can say that monomer is an amino acid itself and the polymer is a protein. So amino acid, there are 20 kinds of amino acid that differentiate by the R chain here. There is an amine chain and the carboxyl chain here, and the R can be anything. So it can be a non-polar, it can be uncharged polar or a charged polar. So amino acid itself can polymerize to form a linear chains, for example, like dipeptide here at the bottom one. So these two amino acid are joined together with the peptide bond here. So it can be said that this is a dipeptide. So moving on, how to quantify the protein? So a protein quantitative analysis, there are three types of it. So there is a volumetric and a gastometry and spectrophotometry. And the volumetric analysis containing there is a Hall, which is the determination of the total N, and then formal titration there. So they are using the alkalimetric titration, and while the gasometry analysis, they use the converting the amine group to an N gas. While the spectrophotometry here, so there are two types of spectrophotometry, the UV and the visible light. The UV is used for quantify the proteins with aromatic groups such as phenylalanine, tyrosine, and tryptophan, while the visible light such as here burette, folin, and laurie. In the in our experiment here, we are going to use the visible light spectrophotometry. We are using the Biuret analysis. So the Biuret analysis is measure the protein complex between the peptide chains that making a complex with the Biuret ion such as Cu2 plus here, and their protein complex between the protein and Biuret reagent here, Cu2 plus, will be performed in a blue-purple color, which can be measured under the wavelength 550 nanometer. So the Biuret reaction gives a plus reaction to a compound containing CH2NH2 or CHNHNH2 or CSNH2. The advantages of using the Biuret method is that the concentration of a protein is comparable to the polypeptide bond, and then the method is simple and fast. While the disadvantages of the Biuret method here is they are less sensitive, and then the color is sometimes unstable. Yeah. So why this method is less sensitive to others? Because many sample that containing like this here chains CH2 and several this for this kind of chain, so they can be measured by this method. So it can be protein, it can be others sample, so that's why it's less sensitive. So moving on, why a big questions, why we need to measure the protein concentration and why we need to measure the enzyme activities. Is that really important? So for our experiment, we're going to measure the GST enzyme. So as you can see here in the picture, the phase 2 enzyme reaction, there involve three different enzymes such as glucuronidase or UGT in glucuronidation, while the second one is enzyme sulfotransferase or ST involving in sulfation, and the last one is enzyme glutathione S transferase involved in glutathione conjugation. So we're gonna explain about the GST here or glutathione S transferase. These phase 2 enzyme reactions are involved in the excretion of some xenobiotics in our body, so including the GST1. So as we can see here in the picture, the glutathione and xenobiotic helped by the enzyme of GST will transform or make glutathione as conjugate to be excluded from our body. For the glutathione conjugation, as I already explained to you, that there are three enzymes there. So the glutathione conjugation include several xenobiotics such as electrophilic xenobiotics or the xenobiotics that already transform biologically to electrophiles. While the substrate for the GST itself share three common features, such as the substrate must be hydrophobic and the electrophilic and react to non-enzymatically with glutathiones at a measurable rate. And so moving on to the next slide, there are three types of GST conjugation, or there is three different processes for making the glutathione as conjugate or conjugation. The first one is direct conjugation by displacement of an electron withdrawing group. So sorry, it's covered by this screen here. So for example, there is a 4-nitroquinoline 1 oxide here on the left. So this nitroxide group will be displaced by the GS minus 1, so that this position will be displaced by the GS. While the direct conjugation by adding of glutathione here means that, for example, the diethyl malate here, the H plus ion or the double bond here in the C will be adding or will be broken down by the GS, so the C double bond will be replaced by the C with the GS ion here. While the conjugation of a strain ring system formed metabolically. For example, this is a chlorobenzene, or P450 will be formed or will be transformed into 3,4 oxide, which is structurally this is more unstable by P450, so that the Gs minus or glutathione minus here can be displaced or can be replaced the H ion here by the Gs ion. As you can see here, there are several types of Gst detoxification, or types. There is alpha and phi and mu and theta and phi and zeta and so on. And the substrate and the Gst enzyme firstly published in 1974, including the CDNB that you are going to use in your experiment. So moving on, the preparation upon determination of enzyme specific activity measurement. So in this experiment, you are going to do 5 different things, or 5 related experiment. The first one is prepare the cytosolic fraction containing the Gst enzyme from mice. This is already prepared by by our staff. And the second one is the determination of enzyme activity, or Gst, in a rat liver by the CDNB method. And the third one is a preparation of bovine serum albumin standard curve using the Biot method. And the fourth one is determination of the protein content of cytosolic fraction containing Gst by the Biot method. And the last one, you're going to determine the enzyme specific activity calculation. So overall, there are 5 steps to finally can measure the specific activity of the Gst enzyme. So for the first preparation, cytosolic fraction of the Gst from mice, if we can read here, we have we need to feed the male rats, which is the strain Sprague Dolly 1, with the weight 200 and up to 250, with a pellet and water until it's full, and let the rat fast for 24 hours prior to the next step. Then after that, you need to sacrifice the rat with the neck and with the neck dislocation. And for the cytosolic fraction preparation containing Gst by a centrifuge method. So after you did the dislocation, so the liver will be chopped up, and then will be separate between the enzyme and the other fraction by using the centrifugation method. And then after that, take the rat liver and immediately put an phosphate buffer in a cold one, and at pH 7, then measure the weight, and crush the rat liver in a phosphate buffer using a cold blender for 5 minutes. And after that, centrifuge the refined homogenate at 10,000 G for 30 minutes at 4 degrees. Take and transfer the supernatant to a new tube and discharge the precipitate layer, and centrifuge the supernatant at 105,000 G for 90 minutes at 4 degrees. It's like several steps of centrifugation to take the Gst enzyme. And the supernatant layer is cytosolic fraction containing the Gst enzyme, as I already explained before. After the centrifugation, there's supposed to be 2 layers there, so here we need to take out the supernatant and store the cytosolic fraction at -80 until further use for the CDNB conjugation with GSH. And the first step that already prepared by our laboratorian. So the second step is we're going to determine the enzyme activity Gst rat liver by the CDNB method. CDNB is stand for 1-chloro 2.4-D nitrobenzene. So here, as we can see on the table, there are two different fraction or two different type. One is a Gst cytosol fraction. One is an aqueous, or it's distilled water. So why we need to use the distilled water? Because it's going to be used as a blank here. So please prepare two types of flask there. As you can see on the explanation of the experiment, you are going to prepare two flasks. One is cytosol fraction, and then the other one is distilled water, and put 920 phosphate buffer, and then put 20 microliter of GSH and 20 microliter of CDNB, and then the last one is add the cytosol fraction for sample and distilled water for blank. The Gst conjugate, after you mix it up all of this reagent, you will have a Gst conjugate product form, and then measure at 340 nanometer from minute 0 to minute 3 using a spectrophotometric as a simple kinetic program. So it means after you mix all the reagent there, so you need to measure the 0 minute and minute 3. So you have two different measurement result for each sample. So you will have four different measurement there, two for sample and two for blank. So please make a note that the measurement time, which is considered as the 0 minute, the same as the other treatment. It means that if you are mix all the reagent, so the 0 minute, it means the last time when you added the CDNB for the sample, also similar for the blank one. So starting time is calculated, yeah, after you add the CDNB. So the CDNB one should be added the last after you put all the reagent there, such as first cytosol fraction, phosphate buffer, GSH, and the last one you need to put the CDNB and make it as a 0 minute. So the end of product will be rate or absorption per minute. So after that, the Gst enzyme activity is then measured in micromole product per minute. So the observation you need to measure there is reaction speed. So it means the absorbance, the delta absorbance. So so as you can see here, there is a two measurement, the 0 minute and the minute 3. So the delta minute, it means that the minute 3 minus minute 0, so you can get the delta absorbance there per minute. And then the other calculation here is you need to calculate the Gst activity and speed reaction in B here, divided by 8.500 micromole per micromole centimeter. So you can get the micromole GSDNB per minute is stated as C. The result of this calculation is needed for further calculation the activity of the enzyme. So you need to measure all of this step. And the third one, after you measure the speed of reaction, so the third one is making a standard curve. Why you need to make a standard curve? Because you need to measure the concentration of the Gst enzyme. So here is how to measure how to make the BSA standard curve, or BSA stand for the bovine serum albumin, using the Biuret method. So the material is Biuret reagent and the standard solution of BSA, and the phosphate buffer you can read by yourself. And then the procedure is very simple. So weigh the albumin and put it in a volumetric flask and add a little aqueous, and make an albumin standard curve with the series concentration ranging from 100 to 700 microgram per milliliter. So the procedure in C here is very important because you need to make a range of concentration that you're going to use to make a standard curve. So here it is, the standard curve table here. So there is 1 up to 6 and the other one sample, so it's going to be a 7 different flask or 7 different solution you need to prepare. The first one is albumin solution, which is contain the BSA 1 microgram per milliliter, and the second flask is the BSA concentration of 1.5, and the other one is 2 and 3 and 4. And for the last one, there is no BSA there because it's going to be your blank. And then the last one is your sample of GST sample that you are going to figure it out how many concentration is there. So the standard curve will be making this six absorbance reading, and you need, after you read the absorbance there, so you need to make a linear graph equation like this is the concentration, this is the absorbance there. So you're going to find out the concentration of the sample you are you are going to measure. So for example, or after you mix all of the reagent to make the standard curve, and then you got the equation, like for example y equal to bx plus a, and for example if the absorbance is y, so if the absorbance is 0.234, then the 0.234 equals bx plus a, and the x1 stand for your concentration, your sample concentration. So it means that 0.234, which is y minus a, divided by b. So the fourth step is the determination of protein concentration of cytosol fraction containing the GST. So here also all you need to do is just mix all of the reagent here, cytosol fraction, phosphate buffer, and biuret reagent, and incubate those solution in the room temperature for 10 minutes. And measure the absorption of the mixed solution at 550 nanometer against a blank, which is containing no cytosol fraction. And then after you read all of these absorbance here, this absorbance here, you're going to measure the cytosol protein content, which is the total volume per cytosol fraction milligram per microliter. And this one is, you can get this one by measuring this one first, and within the 4 microliter there will going to be some milligram protein. So it means after you measure the x protein concentration, which is shown here, you can put the concentration as a cytosol protein contained by dividing the 1001 times the total volume divided cytosol fraction volume. And then within the 4 microliter there are going to be D, the cytosol protein contained within the 4 microliter. They're going to be a milligram protein. So please make a note, 1 unit of enzyme is the total of enzyme that catalyze 1 microliter of substrate reaction per minute, and an enzyme specific activity is the total of enzyme units per milli divided by the protein concentration in milligram per milli, or the total enzyme unit over milligram protein. Therefore, the unit for specific enzyme activity is unit per milligram. And then the purity of enzyme is proportional to the value of its specific activity, meaning that the greater the value of the specific activity, the higher the purity of the enzyme. So the next question is why we need to measure the enzyme activities regarding to the pharmaceutical field. Why measuring the GST enzyme is so important. For example, here there is one scientific report with the title Advance in Anti-Tumor Agents Targeting Glutathione as Transferase. So the GST is, as already explained to you, that GST is a family of phase 2 detoxification enzyme. So this enzyme catalyzes the conjugation of glutathione, including the chemotherapeutic agents. So the GST is an enzyme that will be exclude or catalyze will help the excretion of some xenobiotics. So the GST in cancer patients, that as you know that the cancer is such a cell which is have a super growth, that they can divide, the cell can divide into a multiple or multiple produce multiple cell. So you can imagine that the GST enzyme is overproduced there. So it means the excretion of some xenobiotics will be totally also multiple times doing excretion. So it means that whenever the cancer patients they receive some chemotherapeutic, so it means the cell will be excrete or will be exclude the chemotherapeutic drugs also in huge amounts. So the GST enzyme uh, can be targeted as an xenobiotic target. So whenever the GST is inhibited, so it means that the chemotherapeutic drug can be used or can be treated the cancer patients successfully, because the GST is inhibited and the GST cannot express or exclude any chemotherapeutic drugs outside our body. So it means that the chemotherapeutic drugs can give a benefit, a maximum benefit to our body. So thank you for listening.